Hi, Dr. Brian Koffman here at ASCO 2014. On the last day, I'm here with my doctor, Dr. John Byrd out of Ohio State. Dr. Byrd, do you want to introduce yourself, please?
Yeah, hi, this is Dr. John Byrd. I'm a professor of medicine and director of the Division of Hematology at Ohio State University.
Dr. Byrd, I just heard you present a paper commonly called, I think, the RESONATE trial, and this is a Phase 3 trial. Maybe you could talk to us about, I think, the difference between the Phase 3 trial and the Phase 2 trial in terms of what kind of information you can tease out of that?
Yeah, absolutely. Well, it was literally, Brian, the last thing at the meeting, and we saved the best for last for our CLL patients in that, this was the first randomized Phase 3 study of ibrutinib as compared to a comparator treatment, ofatumumab. And the beauty of Phase 3 studies are that they allow us to understand what things are associated with new drugs versus what is associated with the natural history of CLL, because you follow patients. Patients are assigned to one therapy or the other. And we learned a lot in this Phase 3 study. We obviously learned that ibrutinib was a much better therapy than ofatumumab. It beat the pants off of ofatumumab for all the clinical measures: response, time in remission, and overall survival. Importantly, what we saw was when you followed patients on each of the arms, that patients stayed on ibrutinib longer, and when you adjusted for the time for patients being on therapy, the concern we had that ibrutinib might cause serious infections, actually what we saw was that the risk of infections was lower on ibrutinib, at least for major infections. We saw some new side effects with ibrutinib that were not seen before, previously. One called atrial fibrillation, where the heart gets into an irregular rhythm. And in most patients that was manageable, but that's something that's going to have to be monitored in the future. Many doctors and patients alike were worried that ibrutinib may increase the risk of major bleeding or Richter's transformation. And the beauty of a Phase 3 study is, we saw that in both the arms, say that the frequency of Richter's transformation was the same. There were two cases, two unfortunate patients in each arm that developed that. And while there was some minor bruising, there was a minor bruising and ecchymoses in the ibrutinib treated arm. There was this frequency of serious bleeding was the same. The trial also told us a few things about ofatumumab that we didn't know. In our own study that we did with combining ibrutinib with ofatumumab, we saw peripheral neuropathy, and there was a question of whether that was an interaction between the two, because that had never been something that we had seen with ibrutinib. And in this randomized trial, we saw that actually it was the ofatumumab, and this had been missed. So, ofatumumab does appear to cause a sensory neuropathy, and that's generally where people have numbness around their lips and sometimes their tongue and their fingers, and that typically goes away with time. But again, it's something that we learned about the study. We learned about ofatumumab through doing the Phase 3 study.
And could you tell us a little bit about what you were able to find in terms of the efficacy data, which I understand was pretty powerfully in favor of the ibrutinib?
Yeah, the efficacy data was presented both from the Phase 3 study, where we saw that patients had a much higher response, upwards of 85%, as assessed by the investigator. Whereas it was much lower with the ofatumumab, and more importantly, 86% of the patients who were randomized to the ibrutinib remain on the ibrutinib. So the progression-free survival was at 6 months was 88% versus 65% with the ofatumumab treatment. The survival was longer. 90% of patients were alive on the ibrutinib arm versus 81% at 1 year. And that's despite this was a very contentious trial in terms of, as the data got much better with ibrutinib in the Phase 2 study, patients and their doctors really wanted them to be able to get access when ofatumumab stopped working. And so the study was modified in the middle to allow patients who had progressed through ofatumumab to go on to ibrutinib. and so we still saw a significant survival advantage, which again really hits home how much better of a drug ibrutinib is than ofatumumab for relapsed CLL patients.
And this hits home another kind of side issue, because, I mean, it's good that that showed, but in other ways it's pretty sad that it showed, because that survival advantage was at the cost of those patients who couldn't cross over. So going forward, how does one, with these promising drugs, how does one build that in and still get a drug to be approved? I mean, this is your life, is designing these trials. From a patient's perspective, I want the crossover in there from day one, but I understand that the regulatory authorities may say, hey, you can't prove a survival advantage if you don't do that. So help me understand that.
Yeah, well, it's a complicated issue, because there are a lot of drugs that are not very good. And the regulators are trying to protect patients from getting things that aren't good. And so we've been having a lot of discussions at Ohio State, and I think one of the things, and this hits so close to home, because the first patient that went on the RESONATE study was a patient of mine that got ofatumumab, randomized to that, and didn't respond and died of pneumonia before he could get ibrutinib. And he's a very sweet man, and so and I think all the doctors that participated in the study wanted a crossover. And an idea that we have is, but you're right, the regulatory agencies, particularly in Europe, and with companies develop a drug, worldwide, they have to consider what everybody says. And it's not just the U.S., it's Europe. And so, a design that we've been thinking about for drugs like ibrutinib is an adaptive design, and that was sort of applied in this study. By the time we got to August, to August of 2013, we really knew the responses with ibrutinib from the phase 2 study were very, very durable, and we probably knew that sooner, actually. And we were able to convince the regulatory authorities at that point that a crossover was going to be necessary, not to compromise the study, because if the drug got approved, patients were going to go on the drug, and so I think designing a study where, if the phase 2 data continue to improve and look very good, that prospectively, as the study is designed, there's a crossover written in, or alternatively, you just have a crossover. Personally, as a physician, I'm always in favor of crossover, as long as you have a good drug that doesn't bring risks to patients.
Talk to me a little bit about the group. It was a small group, but the groups that you did see progress, what their cytogenetic characteristics were. I think we can anticipate what they were, but outline who is at risk and how big that risk is of progressing.
Yeah, the RESONATE study that was presented here has short follow-up, and so really the patients that progressed thus far have mainly been Richter's transformations patients. Brian, you're aware, so our group, because we've treated a lot of patients, and we've worked collaboratively with the group in Germany, with the company Rich Furman at Cornell, in getting samples from patients who had received ibrutinib and who progressed, and studied them, and then we've extended experience at OSU, and essentially the collective experience, and Dr. Woyak presented a poster on this and has a paper in the New England Journal of Medicine with work that she did with Dr. Johnson and others, showing that most patients who progress within either the first 12 months, and that group of patients is with Richter's, and then after that 12 months, a subset progress with CLL. And it's about 2/3, 1/3. Almost all those patients have deletion 17p, or complex karyotype. And Dr. Woyak actually showed complex karyotype and BCL6 overexpression, and there was a trend toward number of prior therapies was most predictive of who's going to progress. I think what's encouraging for our CLL patients is we have a blood test that where we can identify. There's essentially two common mutations that occur. One where the cysteine, the cysteine site where in BTK where the drug binds, and then a downstream kinase called PLC gamma 2. And so you can look in most patients who relapse on ibrutinib with CLL and find these, these mutations. And there are drugs that are coming forward that specifically target at least the common BTK mutation. I think as we move forward with ibrutinib, say particularly in patients who are at high risk, we'll be doing monitoring for resistance, similar to what we do with CML, where then if we see a resistant clone coming out, we'll either be changing drugs or adding something to try to get rid of that that mutational clone.
So are those tests that you could be doing before the patient relapses to say that you're at risk for relapsing, or there are other markers that you can look at to say, okay, I see a relapse coming? How does that present in the ibrutinib case? Yeah, so there's a test that's predictive, that you could do at baseline, and the FISH test, so the 17p, the complex karyotype, those are going to be genetic abnormalities that are present before starting therapy that predict that you have a higher risk of relapsing. Monitoring during therapy, you're looking for a genetic or a mutation, the BTK cysteine to serine 41 mutation, that when it shows up, it's relapse is going to come. It's just, and typically when you see that, you can see it before patients manifest clinical relapse. Sometimes you can see it 3 to 6 months earlier, and the clone will increase in time because the ibrutinib doesn't get rid of it, and it sort of has a selective advantage to grow. The drug gets rid of the CLL that don't have it, but the CLL that does have it, continues to grow. Is that a test that you're doing experimentally at this time at OSU? Yeah,
so it's a test that we're doing experimentally, and we've got a very sensitive assay, and we're in the process right now of moving it to our pathology lab where it can be done and reported back to patients. Now we're just doing it in an ad hoc way where we're using it for research, but we plan to move this forward and hopefully over the next 2 to 3 months have it up where we can actually do it and report it back to patients and to their doctors.
All right, let's take a quick break here and we'll continue in a minute.